Good afternoon, everybody. It's my pleasure to present Professor Dr. Hugo Barrera, and I want to talk about some important things of his extensive trajectory. Professor Dr. Barrera is doctor in biomedical science from the University of Texas at Houston, post-doctorate at the Institut deGénétique et Biologie, Moléculaire et Cellulaire of Louis Pasteur University in Strasbourg, France. Professor of Molecular Medicine at Universidad Autónoma de Nuevo León, Instituto Politécnico Nacional, and Tecnológico de Monterrey. Science and Technology Commercialization Specialist from UT Austin and Clinical Researcher Specialist from Tecnológico de Monterrey. Professor Dr. Barrera is a Mexican biologist, biochemist, researcher, entrepreneur, professor, and academic, an author of several studies of molecular biology, epidemiology, and biomedicine. He's a precursor of molecular biology in the Universidad Autónoma de Nuevo León in Monterrey, Mexico. Creator of the graduate programs of genetic engineering and molecular biology, and of biotechnology genomics of Universidad Autónoma de Nuevo León and Instituto Politécnico Nacional, respectively. Also of the Genomic Biotechnology major of the School of Biology and of the Technology Transfer Center in Universidad Autónoma de Nuevo León. Author of more 200 peer-reviewed publications with more than 3,000 citations through international biomedicine books to national educational books. He's member of the National Researcher System Level 3, and he founded the Unit of Engineering and Genetic Expressions Laboratory. In 2019, Professor Dr. Barrera received the National Science Award by the Mexican government. Professor, it is a pleasure to have you here.
It is my great pleasure and honor to be here, and I would like to thank the organizers for inviting me to this meeting, especially the president of the meeting, my dear friend, Patricia Zambrano, who, by the way, has done an outstanding job in our university as the General Director of Research. Congratulations, Patricia. I also want to thank the whole team of organizers for carrying out this Congress in the middle of perhaps the worst health crisis that our generation will face. Because of that, because of the nightmare that we are experiencing, I have chosen to speak about biomaterials. Because I would like to convince you of the important role that biomaterials have. Let me start by saying that the actors of the story that I will tell you are precisely the biomaterials, which means the DNA, which is here, the RNA, and the proteins. They will be the players of the whole history and all the tales I will give to you today. And I will also speak about the advancement of science using biomaterials to produce new drugs, new medicine, including vaccines. And finally, I will touch on the nightmare, on the health nightmare that we are facing, the virus, the coronavirus pandemic that we have. This is the outline of my presentation, and let's get started. I'm sure that you experts in materials probably have your favorite cosmic phenomena, those described here: black holes, quasars, pulsars, supernovas, and so on. These wonders of the universe indeed can impress us. But I will try to prove to you that even more impressive than these wonders of the universe are the biomaterials. Let me tell you why. And basically, because to be aware of the existence of these wonders of the universe, we need to be alive. We need to have a mind and be aware of the universe itself. And precisely life was made possible in our planet thanks to the biomaterials that I described to you. Regarding beauty, to me, the most beautiful is DNA. And regarding complexity, I'm going to give you an example. These biomaterials, which I say before, they made possible life on our planet, they are capable of doing extraordinary things. For example, the ribosome, the small organelle in the cell, capable of producing all the proteins that the cells need. But let me give you an idea of how important, how, I will say, precise is this little machine. If one were to synthesize a polypeptide, a protein, made of the 20 different amino acids that most proteins are made of, and perhaps the length will be 200 amino acids, this protein, it will be 200 to the power of 20. The possibilities of creating all these different proteins from this pool of a 200-long polypeptide is larger than the number of stars in the universe. And the time to build a protein will take an eternity. However, this ribosome makes proteins in a matter of a second and with extremely high precision. By the way, proteins are the workers that keep the cells moving. So some of the most important elements in the cell are precisely the proteins, and the ribosome synthesize them. How we do it? Basically by taking genetic information from the messenger RNA. And messenger RNA will come one after the other in my talk. You will see it very frequently. You will hear it very frequently. So by using this template with the genetic information, this machine is capable of synthesizing, I say, a protein anytime it's needed, in the amount it's needed, and precisely with the sequence that is needed. It's a miracle, and that miracle makes life possible. The current view of the origin of life in our planet calls for the existence of an RNA world. RNA can easily be made from nucleotides. RNA can replicate itself. And RNA can catalyze the synthesis of proteins. So it is believed when the catalytic activity of RNA was discovered back in the 80s of last century, is when the idea came that perhaps the first genetic information that made possible life was RNA. Later on, RNA was replaced by DNA as the reservoir of genetic information. It has many advantages over RNA. And even later on, RNA was replaced by proteins to carry out the cell functions of the cells, to catalyze the cell functions of the cells. So the three biomaterials I'm speaking of are precisely DNA, RNA, and proteins, and they make life possible in our planet. So the current view of the genetic information and the flux of information from DNA to RNA to proteins is described here. DNA stores instructions for protein synthesis, and these instructions are in the nucleus. They are kept in the nucleus of cells. mRNA is like a piece of bits instructing the cells to synthesize proteins. And proteins, of course, are performing the basic functions of the cell. So this is how biomaterials make possible life. I started to train myself as a biochemistry in the University of Nuevo León many years ago. And I started by precisely isolating mRNA. In fact, I isolated total RNA from human placenta. And I will tell you a nice story. I was in this maternity hospital in Monterrey with a pocket of ice with me next to the future father. And the father was walking around waiting for news for the baby. And he will say to me with strange eyes, "What is this guy doing here?" And I was just sitting there with my eyes on pocket. When they announced that the baby was born, he was running from this door, and I was running from the other to get the placenta. So from total RNA, this is electrophoresis, gel electrophoresis, separation of total RNA. I isolated mRNA, messenger RNA from placenta. Then I used a cell-free system for translating the RNA into proteins. And this is a protein gel. used to find out that the most abundant protein that mRNA from placenta codes is a hormone called placenta lactogen. So one of the most important proteins made by the term placenta is human placenta lactogen. Then I was able to acquire recombinant DNA skills. And with this technology, we can clone genes. So I cloned the gene for human placenta lactogen. And at the same time, I cloned the messenger RNA for human placenta lactogen. When I hybridized the gene to the mRNA and look at the microscope, we discovered that there were 4 introns in the gene. That is to say, the human placenta lactogen gene is split by 4 introns. Then we discovered that the human placenta lactogen gene was a member of a gene cluster, 5 genes. The human growth hormone gene, and all these genes which are expressed in the placenta. By the way, this is expressed in the pituitary gland. So you have here a genetic system and a genetic model to study, very interesting. Very close in the genome, in the human genome, there are these 5 genes. One is expressed in the pituitary gland, and the rest are expressed in the placenta. How? They are extremely similar. They are close to each other. Why they have these differential tissue expression capabilities? That was one of the mysteries I wanted to solve. So I went back to Mexico. I settled down in the School of Medicine, established my own laboratory, and trained in Army. The Army have the idea of explaining what is a gene, how does it work, how is it controlled, and finally, what use can we make of genes? What can you use them for? These were our dreams, our ideas when we started in Mexico, our own laboratory. Well, we solved the enigma. We solved and we got the explanation why this gene is expressed in the pituitary gland and why the rest are expressed in the placenta. How? We took the pieces of DNA which are in front of each gene in 2 different lengths or versions, the long promoters and the short promoters. Small pieces or long pieces of information in front of the genes. We introduced the short version of each of the gene promoters into cells. And yes, the genes express all in the pituitary gland, all in placenta, but they are not expressed in liver or another tissue, pancreas or whatever. But this is not the complete solution, because as you can see, the pituitary gland expresses equally to the placental genes. The pituitary gland expresses almost equally to the placental genes. So when we took the long piece of DNA in front of the genes, voila, we found the solution. We found the answer. The long pieces of DNA in front of the genes keeps this gene active in the pituitary gland and inactive in the pituitary gland, the rest of the placental genes. And these long pieces of DNA keeps the placental genes active in placenta and represses the pituitary growth hormone gene. So we have solved the problem. We were very happy. And we were even more happy when we were invited by colleagues from Genentech in San Francisco and other places in the world, but basically from San Francisco, we were invited to sequence the entire human growth hormone locus, which we found to be in the long arm of chromosome 17. So we sequenced 60,000 nucleotides. It took us years to do that because it was early in the sequencing technology. Now you can do it in a matter of seconds. But it was taken as a world record. I was very proud to be part of this world record. We had written 1 page of the human encyclopedia. It's only 1 page. It is related to the growth hormone and placenta genes, which I described to you. There are perhaps another 30,000 pages. Genes for the color of eyes, genes for diseases, for everything, genes to construct a human being. But we've discovered that the one that dictates the production of in the pituitary growth hormone and the placenta or placenta lactogen genes. Science took this publication of us as a proof of concept that eventually the human genome will be sequenced, which happened a few years later. So that's about science. What I just told you is about science. But I was asked to talk about science, technology, and society. Let's move into technology. I recall you, these are the biomaterials I'm speaking of. And I will recall you again, this is the one page that we wrote of the Human Genetic Encyclopedia. What can you do with that kind of information? Especially when I was working in a medical school with a university hospital next to it, and they have everyday problems. They were not dreaming in advancing science, but solving the issues that were in front of them. And one of the problems the pediatricians had was to explain this. Why are there children that are born and have growth retardation? Not only that, why are children with growth retardation that even though that you can deliver or administer biosynthetic growth hormone, they don't respond? Well, it was well known that there were these children with growth retardation that even if they receive recombinant growth hormone, they will not respond. Why? Well, we took the information in this one page of the Human Encyclopedia that describes how to produce growth hormone and created a PCR, a polymerase chain reaction methodology to amplify the 5 genes which I just described to you. We amplify them. There are 5 different amplification products. We cut them with an enzyme called restriction enzyme, ACC1. And of 10 patients that were sent to us from the pediatrics department, you can see the last one is missing these 2 bands, which precisely are the bands corresponding to human growth hormone gene. So this kid was lacking the human growth hormone gene. Since he was lacking this gene, her body, because it was a girl, her body never produced growth hormone. So even if you inject them with human growth hormone, for her, it will be like an antigen, and it will produce antibodies and neutralize the treatment. So we created the first, we call it world companion diagnostic, the first companion diagnostic of the genomic era. We were very happy with that because we can help the pediatricians and the children. And we were able to translate genomic knowledge into the diagnostic laboratory. But then we say, if we have the gene for growth hormone, can we produce growth hormone? Because Mexico needed growth hormone. So we tried. Together with my colleagues in the laboratory, we introduced the gene of human growth hormone into yeast. And we found out that when you do the fermentation of these yeast, they secrete a band of the size expected for human growth hormone, recognized by antibody against human growth hormone. When sequence is identical to pituitary growth hormone, when you test it in the lab, it has biological activity, and because of that, we got a patent in the United States, Europe, Japan, and Mexico. And we licensed the technology to a Mexican biopharma. So again, we proved that we can translate genomic information for diagnostics and for developing new drugs. And another member of my team have this dream. Can we create viruses that enters normal cells and they do nothing? But when they enter tumor cells, specifically tumor cells, they will replicate and kill tumor cells. Can we make viruses that selectively replicate in tumor cells? We tried. We took adenovirus that we know that has a piece of DNA in its genome that dictates the production of a protein that triggers replication of the adenovirus. And we say, what if we replace this piece of DNA of the adenovirus by that similar piece of DNA from human papillomavirus. So it will be a hybrid between adenovirus and human papillomavirus. You probably know the human papillomavirus is the cause of cervical cancer, a huge problem in Mexico, Central America, and many countries of the South Hemisphere. So the idea was, if this recombinant, then the recombinant will be, the replication of the recombinant will be triggered by a protein for the human papillomavirus. And that will lead to the replication of the virus exclusively in the cervical cancer. Well, we tried that in an experimental model, and we were surprised with the results. We have this mice model in which we implanted cervical tumors in both sides of the back. You can only see one here. You don't see the other one. We injected the recombinant virus in this tumor, and we noticed that the tumor was decreasing in size, but to our surprise, the non-injected tumor was also decreasing in size. So the virus escapes from the site of injection, goes to the other tumor, and also kills the other tumor. So we were very happy. We went and we published this paper. We tried to patent it, and I was very busy doing other things, and it was patented only in Mexico. Can you imagine? Only in Mexico was patented, still haven't been patented in the rest of the world. Be careful of the lawyers. But in any case, we tried to produce the virus in Mexico, and we didn't find an incubator, a bioincubator, a place where to do with good manufacturing practices, what is called GMP practices, the virus in Mexico. And we needed to go someplace in the world to manufacture the virus, and of course, money and other limitations were in our road. But people noticed that we were working with gene therapy in Mexico. And colleagues from the University of Texas, specifically from Texas Children's University, Texas Children in the University of Texas, invited us to start a gene therapy against cancer, in this case, prostate cancer, to start a gene therapy against prostate cancer in Mexico. It was amazing because we invited professors from the university if they wanted to be guinea pigs for this project. And many say, yes, we can try that. So we had 10 professors from the university diagnosed with early stage prostate cancer that came to help us. We got the green light from the ethics committee, from that research committee from our university hospital, and we injected into the prostate of these professors the virus, the recombinant virus. This is an adenovirus, or was an adenovirus, carrying a gene from herpes simplex virus, another virus, thymidine kinase, that's the name of the other gene. This thymidine kinase gene, of course, produces the thymidine kinase protein, or enzyme. And this enzyme, this viral enzyme, is capable of taking a nucleotide analog and introduces it into the DNA and stops replication, and the cell dies. It's a suicidal gene therapy. That's the name of this technology. But the ethics committee say, after you play with your technology, you say it's very safe and perhaps very promising, you have to remove the prostate from the professors because surgery is the only thing that cures. We don't know about your technology. So we were very lucky that we saw that the biomarker for prostate cancer decreases in all the patients that we injected. And when we had the prostate removed by the surgeon in our hands, we carry out a lot of experiments, and we proved that the virus was there, thymidine kinase enzyme was there, necrosis was happening because the cells were dying, and the immune system was invading the tumor. That was done about 16 years ago, and it has been probably the only gene therapy protocol ever done in Latin America. We were also very happy with this achievement. And then I was in France visiting my professor in Strasbourg, Professor Chambon, and he invited me to visit one of his companies. Transgene was the name of the company. I went to visit Transgene, and they told me they were trying to do a vaccine against cervical cancer, but that they were trying to find patients in Europe, and it was very hard. And I very arrogant say, well, you can come to Mexico, we can do this protocol right away. I went from there to visit another colleagues in France. And when I was back in Monterrey, my secretary told me, Dr. Barrera, there is a French woman waiting for you in your office. It was the director of the transgene that said, well, you say that you were going to help me. I'm ready to start. So we started a protocol against cervical cancer with the help of transgene, well, we will help in transgene, and indeed, we finished the project in Mexico faster than Europe. We didn't publish that because there was, of course, a non-disclosure agreement. So as you can see, we can use biomaterials to program cells, yeast, human cells, the body, to produce new medicines. But then we were very happy, and the pandemic came. Came from China, from Wuhan, as you already know, at the end of 2019, and it spread around the world very, very fast. We all know now how is the virus, what is the structure of the virus, and we all now know that the spike protein on the outer surface is responsible for entering into the cells and doing what the virus does. We know now how the virus produce this acute respiratory severe symptom, SARS. We know very well all the process that happened. Unfortunately, we don't have a drug, an effective drug right now. We only have some vaccines, and I will describe to you them. But what is a virus? Well, a virus is a small infectious agent, very small, by the way, of very simple composition. You saw proteins and nucleic acids. And it comes from the Latin poison. That's what they mean. There are several hypotheses of how the virus appeared on our planet. One says that there were cells trying to become real cells, and they decided to be parasites. They never became cells. The other says there were cells that decided to reverse and become parasites. It was simpler to parasite. And another says there are genes escaping from cells and invading other cells. Whatever it is, what this hypothesis is the correct one, viruses infect all living organisms in our planet. There are viruses that infect bacteria, they're called bacteriophages. There are viruses that infect plants, humans, of course, animals, everything. In fact, these days, 500 years ago, the Aztec Empire was fallen in a battle against the Spanish conquerors. Now we know that it was not the military power of the Spanish invaders, but rather a secret weapon that they even were aware of, viruses. 90% of the population of the city of Tenochtitlan, that was the name of Mexico City 500 years ago, died from the viruses. Measles, and other viruses. Several viruses killed them, okay? So you can see how important and how dangerous are the viruses. Now, we are here all scared of being contaminated with the virus. But we have a solution. It's called a vaccine right now. We would like to have an antiviral product, but we have vaccines. What are the vaccines? Vaccines are a type of medicine that trains the human immune system to fight an invader, a microbe that is invading you, even if you have never seen it before. It prepares you for that. With the vaccine, we train your body, your immune system to recognize fast and attack an invader. And it can be a weakened, killed, or fragmented microorganism, or a protein from this agent, like the spike protein from the coronavirus. And the father of vaccination is considered to be Edward Jenner, an Englishman, that did the following experiment. He took pus from a milkmaid that was infected with cowpox and inoculated a child. The child presented mild symptoms from the cowpox, nothing compared to the smallpox that kills It's very, very dangerous and kills humans. Then Jenner challenged the boy with a smallpox from a patient, and he didn't die. So he proved that you can protect against the disease using a very similar virus. That was Edward Jenner. You can see it here. Well, doing the experiment. And perhaps you don't know that Dr. Balmis from Spain convinced the King of Spain to finance the first philanthropic world expedition of vaccination. He left from the north of Spain to the rest of the world with the cowpox vaccine. How he kept the vaccine alive using orphans. Isabel, that nurse that was the head of that orphan place in Spain, came along. In fact, she stayed in Mexico, and there is a national prize on nursing that has her name. So we don't know how many lives they saved, but you may question ethically using orphans to keep a vaccine and take it around the world. But that was the technology then. There was nothing else. So the history of vaccination is amazing. Perhaps vaccination, together with safe water and even antibiotics, are some of the most important contributions of science to mankind. The technology has evolved. The Chinese in the 16th century were trying to inoculate and protect persons by injecting the human smallpox. Edward Jenner, as I say, used the cowpox. So that Edward Jenner and many others improved the technology, and of course, later on, it came the recombinant DNA to produce the very first vaccines that have never been produced before, hepatitis B, human papilloma vaccines, and another vaccines. So technology has improved vaccination. And now we can say that there are many diseases that are either controlled or even disappear from Earth thanks to vaccination, like measles and polio very soon probably. But of course, there have always been questionings about vaccination. Not now, always. Since Jenner, they say, well, these vaccines are going to convert you in a monster or whatever. That ignorance. There are currently close to 300 new vaccines being developed for allergy, cancer, infectious disease. Even there is something there perhaps for Alzheimer and other diseases. The future is very promising for vaccines. So let me tell you a little bit about vaccines now, because vaccines are putting our society in a huge dilemma. Now let's see what science plus technology is doing for our society. Well, it was amazing what happened last year. In a single year, we were able to identify the gene, sequence the genome of the virus, identify the virus, sequence the genome, develop diagnostic tests, what we call the PCR test, and the fast antigen test, and the fast antibody test. There are tests all over. Produce many experimental protocols to see if this drug or this other drug can help to fight COVID. And even finish the trials for most of the vaccines that now are approved on the Emergency Act. So it was a remarkable year of acceleration of technology and global cooperation. But also, we have already a lot of experience dealing with other coronavirus like SARS-1, MERS, terrible viruses like the Ebola, Zika, and blah, 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 and since the 80s with the HIV virus. So we have a lot of experience in the past doing research. What happened last year is that everything was accelerated because there was global cooperation and funding. By the end of last year, Margaret Keenan received the first jab of Pfizer vaccine. And since then, to you. I'm already vaccinated. I hope most of you have received the vaccination. Vaccines, which the one are being approved, are either inactivated viruses, live but attenuated in the lab, proteins, in this case, spike protein produced by recombinant DNA, virus-like, vector, virus delivering the gene for spike protein, mRNA, DNA coding for spike protein. Most of these are new. Yes. This is non-classical technology for vaccines production. It's done by recombinant DNA. This is a big surprise, mRNA. Well, I'm not surprised because when I was little, I was working with mRNA. DNA. And these are the winners. There are hundreds of vaccines being developed, hundreds. But these are the winners. The Pfizer, Moderna, AstraZeneca, Sinovac, Gamaleya, Johnson, Novavax, blah, blah, blah. And their power, their effectiveness as vaccines is amazing. You can see here the numbers. They protect you from severe disease if you are vaccinated. They don't protect you from getting again the virus if you get exposed yourself and get contaminated. But they protect you about severe disease and about going to the hospital, as many did before vaccination, unfortunately, and many died. But you can see here two. The leading ones are made of messenger RNA. But the pandemics brought many other problems. It quickly filled the hospitals. It quickly claimed all the resources from the health systems of governments. It was a nightmare, a real nightmare. And there was some collateral damage because there was no room in the hospital. Medicine were scarce for cancer patients, for persons with heart problems, and so on. And probably you are aware that in Mexico, there was even a very severe crisis of lack of medicines for children with cancer. So too bad. It is a pity that the academia, the scientists, the technologists, the innovators, we are not working together hand by hand with the government. This is mRNA. You see an illustration. It's a string of nucleotides that instruct the ribosome to produce the spike protein in your body. And who said mRNA is a new technology? That many people that are against vaccines keep telling you. It's an experimental drug. They are experimenting with you. Look at this. The discovery of mRNA was in 1961. And since there until now, there have been many progresses in science and technology and innovation to use this biomaterial called messenger RNA to instruct your body to produce a spike protein, so when you encounter the virus, you are prepared to fight it with your own antibodies and memory cells. How is it done? Well, basically in the lab, they take a DNA that is transcribed in vitro in the laboratory and generate messenger RNA. They put the messenger RNA in a nanoparticle made of lipids, and that's what they inject into our arm. Nanoparticles of lipids with the mRNA inside. In fact, the technology is very powerful. You can, not only this is what happens inside cells, but you can produce artificial mRNA to produce any protein you want. Or even you can produce antisense, the opposite sequence of any mRNA, to block some messenger RNAs in your body. For example, messenger RNA coding for oncogenes in the tumors. You can stop them. I see the future of this biomaterial called messenger RNA as a very bright one. There are applications in cancer, immunotherapy, infectious diseases. You are not seeing that. we are very excited of the possibility to have an mRNA vaccine against HIV soon. Allergy, and so on. The possibilities are tremendous. But, as I say, it's a pity that scientists, technologists, innovators don't work together with the government. And even sometimes the government discredit the academicians. They don't support science enough. They question. They even say that we are selfish, that we don't care. We are insensible. We don't care about the society needs. But this is not new. Antoine Lavoisier said many years ago that this is the real purpose of the government, to make the population happy, wealthy, healthy. That's real. That was said by Antoine Lavoisier many years ago. But the French Revolution decided that he was an enemy of the revolution because of his ideas. And he was killed in the guillotine at the age of 50 years. Terrible. One of the most productive, best scientists in France, they took his head in 1 second. And heads like his appear maybe every 100 years. Terrible. What a professor can do, because I just describe what the scientific community in the world, the pharma, the government are doing. What are we doing besides taking care of ourselves and our dear ones? It's our basic responsibility. Well, Pat is here. I'm well known in the university because I don't follow the rules. I decided to do something about it. And I convinced my friends in the industry to donate materials for the university hospital. Thousands of masks, of covers, gloves, dressing things for COVID. I managed to get these rich friends of mine to donate to the hospital. I also have a private lab. So I reconverted my private lab to do COVID testing, as you can see here. And I'm part of a team in Mexico City with clinicians, and we are testing several drugs. We are just sending a patent to USA, and eventually we will publish results from new drugs, which are promised if they are taken early in the infection. I'm also advisor to the Mexican company that is producing the Mexican vaccine called Patria. And finally, I went to Germany and convinced CureVac, the pioneers of mRNA technology, to come to Mexico to try their vaccine. So I hope I have proved to you that among materials, there are biomaterials. And biomaterials play a key role in the origin of life, in the progress of science and medicine, and they come to rescue from this damn virus. I want to thank you for your attention, for the invitation. Here is my email, and thank you very much. And please respect hygiene measurements for you and for your family. Thank you very much. Ya me pasé.
Thank you, Professor Dr. Barrera. And now I'm going to open the question and response session. If you have some question to Professor Dr. Barrera.
Yes, please.
Thank you, Professor, for your presentation, and congratulations for all the things that you are doing. I used to work with materials, metal materials. I was wondering about the images that you are showing. What is the scale? I mean, microns, nanomicrons, or I don't know.
It's an electron microscopy.
It's
nanometers, yes.
Excellent.
And I'm glad that you asked that question, because when I was studying molecular biology in Texas, some of my friends back in Mexico, they say, what are you doing? Genes even don't exist. They don't exist. So I brought the picture. Here is my gene, taken by the electron microscopy. Thank you for your question. Any other question? Please, feel free, there.
Hello, thank you for your presentation. I was wondering of what do you think about the role of patents in the world of vaccination? Should they be patented or should they be free for anyone that can make them?
That's a very good question and very difficult to answer. We all know the patents is an incentive for the progress of innovation. It's an incentive for taking risk, for taking a technology that you have a concept and you may have even a prototype, but you know if it's going to work in the market. So you have to invest your money. And the patents is the governments grant you a patent so you can try to recover your investment. So that's the way they work. Of course, like in this crisis that we are facing, I think it's the responsibility of government, which they are doing, to cover the expenses of everything, to invest in development of technology, and as they are doing, to pay for the vaccines. We receive the vaccines free. We don't pay royalties for the technology. And I will say that the vaccines are quite cheap. They are in the order of $10, something like that, you know. You go to a restaurant and you spend more money for that, you know, and this can save you your life. So in summary, I think patents are needed to continue stimulating the progress of innovation and develop of new products in the market. But in the cases of crisis like this, government has to do something about it. Thank you for your question.
No, thank you.
Any other question? Here.
I would like to ask, I mean, it's probably private information on the subject that you're developing the Patria vaccine. I would like to ask if there's any sort of nanocarriers being developed there, or if there's any use of nanotechnology or nanomedicine, because you mentioned solid lipid nanoparticles in the Pfizer vaccine. Is there something similar in the Patria vaccine?
Yeah. There are two types of vaccines. The one made of, I will go to the extremes. The one that was made with messenger RNA and perhaps proteins. And the ones made either from virus that were killed or attenuated or somehow treated to be less virulent, or viruses that have been modified, like the AstraZeneca, the Russian vaccine, the Johnson vaccine, which are viruses that produce mild symptoms like a flu, but they carry the gene from the spike protein, so they can protect yourself from a more severe virus, more dangerous virus. So the first ones, which are DNA and RNA, they need a lipid mask or cover in order to get into the cells, but because the membrane of the cells is made of lipids, so they need lipids to enter. The other, which are made of viruses, they don't need that. They can enter because viruses, as you saw, are designed for invading cells. And regarding the particles that they use to deliver messenger RNA, yes, they are made of nanoparticles, basically lipids, okay? And in some cases, they put polyethylene glycol and another substance into the product, just to make it more strong, more than anything else. Thank you for your question. Any other question? Okay. They frequently ask me, what is the best vaccine? And as most of the people who are really specialists, I'm not a specialist in vaccines, say the one that you have at your chance, at your reach. And some people ask me, should I use the same vaccine in the second vaccination, or can I change? Apparently there is information that if you have a viral vaccine like the AstraZeneca, then you can use an mRNA vaccine. And we don't know now how long they will protect you. We will need another vaccination in the future. You know, this story is evolving, and we are learning a lot. Patty, thank you very much. Everybody, thank you very much for the invitation. I was very pleased. Thank you.
Thank you again for being here. In name of 29th IMRC Congress, I give you this recognition. Thank you very much.
Thank you very much.
Thank you, everybody, and see you on poster sessions. Good afternoon.